So welcome. Thank you for joining us today. This is a series of 3 webinars, and exciting to have our first speaker joining us from NIST. But before we go to that, I go to the next slide. Some practical things. You will be muted throughout the presentation. And if you have a question, use chat. And there's a box, I presume, in the bottom right-hand side, bottom right-hand corner of your screen with the chat. And then in terms of confidentiality, this webinar is open to both Nimble members and non-members. So please do not post Nimble confidential or proprietary information in the chat or in the chat. At the end, after the presentation, you will receive a survey link. So please help us by filling out the survey. And the reward for that is you would then have access to the webinar recording. So the next slide, John. So this is John's bold title, Big Ideas, Bold Goals, and the Future of Biomanufacturing. And next slide, John. And John, we're fortunate to have John Schiel join us today. John has a Ph.D. in chemistry from the University of Nebraska-Lincoln. And he's a federal technical program manager for Nimble. So he's an important colleague for us at Nimble. And as we work together to build Nimble into successful proof that a government academic industry consortium can provide genuine value to the biomanufacturing industry. John also heads a research lab at NIST, Biomolecular Measurement Division, and he coordinates the NIST MAB programs. So really delighted that you could join us today, John, and share your insight and knowledge and perspective. And over to you. Thank you.
Wonderful. Thank you so much for that kind introduction, Barry, and thank you to all of the Nimble group that has joined us here today. I'm really looking forward to sharing a little bit about the National Biotechnology and Biomanufacturing Initiative and how it correlates to some of the programs that are ongoing at Nimble. So I like the Big Ideas, Bold Goals title. We'll see if we can live up to this very lofty title in the next couple of slides. So to start off, even though we don't have everyone in the crowd today, we're going to do a little bit of an interactive discussion. So I think everyone's going to get the answer to this question correct. So we don't need to all respond in the chat, but this is really a question about perception to say, when we look at this picture, what do we see? And I think the overwhelming response is going to be that we see a pot. So one of the other things, though, if we look a little bit deeper, if we take the lid off and we really inspect this empty vessel, one of the things that we might also see is opportunity. So with this single pot, we can make chili, we can make pasta, we can make mashed potatoes. There are really endless possibilities with this single empty vessel that we can make. And we've been feeding our families and friends for years and years and years with this individual empty vessel. So in our day-to-day careers, we all see another empty vessel. We see a bioreactor. And with that, we also see a lot of opportunity, right? So we see cell therapies, monoclonal antibodies, vaccines, many, many different types of potential opportunities that we can make inside this bioreactor. So it's by no fault that I chose a picture of a scientist here calling one of their friends, asking questions perhaps, working in a collaborative environment, developing this pre-competitive research. And perhaps one of his friend researchers is down the hall at one of the Nimble test beds, and he can come in and help out, discuss, troubleshoot the problem, and come up with a solution. And I think this really is the overarching message of today, is collaboration and working together to solve some of the biggest problems that face the biomanufacturing industry today. So we're going to start by kind of getting an overall gauge setting of the bioeconomy. And I think this is something that a lot of us have heard a lot about recently, of what is the bioeconomy and what is biomanufacturing and how do the two interplay with one another. So I've drawn some definitions here from the NIST lexicon, which is part of the NBBI that we'll talk about today. And the bioeconomy really is any economic activity that's derived from the life sciences, biotechnology, and biomanufacturing. So this can include many different types of products. Pharmaceuticals and biopharmaceuticals, of course, as we all know, but there are also opportunities to make several other commodity materials, plastics, energy, dyes, coloring. So many different types of commodity materials can actually be made and harnessed through the power of biotechnology and biomanufacturing. So biomanufacturing goes into a little bit more granularity to say that we're now using the biological system to produce some good at commercial scale. So I've added a little bit of disclaimer here to also say that this is typically outside of the natural context of the typical biological system, to say that we're not growing corn, for example, for feedstock, or we are growing it for feedstock, for example, but we're not growing it for food to eat as humans or animals. We're actually doing some sort of bioprocess to change these input materials, whether that be feed media or other feedstocks for different areas of the bioeconomy, to transform that into some type of commodity product. And as you can imagine in the bioeconomy, there's impact to several potential global priorities. Health and pandemic preparedness, of course, germane to the audience we're talking to today. But as we've mentioned, there's industrial commodities, there's the ability to produce energy products, and of course, food and nutritional security is something that we've heard recently in the news with the most recent approval of bioreactor cultured chicken. So this really is an exciting time in the bioeconomy, although many of us in the biopharmaceutical space have really been playing in the bioeconomy for a number of years. But as we begin to expand the definition and expand the new types of commodities that can be made with biomanufacturing, it was deemed critically important in the US to lay a strategy for the foundation of how we would actually fund both public and private investments in biomanufacturing, as well as collaborate across government, industry, and academia in really growing the US bioeconomy over the coming years. So the way that we tend to write these national strategies, and I'll talk in a little bit of detail about several of these today, is we actually get ideas from our academic colleagues, our industry colleagues, and we mix them together with our US government perspective to come up with solutions and strategies that will intend to make growth in the bioeconomy. So I'll be talking about the executive order that really launched the entire NBBI, the National Biotechnology and Biomanufacturing Initiative. I'll be focusing on the Bold Goals report that came out in March earlier this year, specifically talking about some of these bold goals that we've laid forward for the bioeconomy. I won't touch too much today on the National Strategy for Advanced Manufacturing, but I'd invite folks to check out this document. It's actually something that covers far beyond bioeconomy and biomanufacturing, but covers every manufacturing space known in the US economy to really talk about advances in other areas like digital, robotics, etc. And you can start to see how many of these new innovations can start to come together to enable new and innovative technologies. So coming soon, I won't talk too much, but I'll give a little bit of a primer. I was hoping this would be out, but it's not quite publicly available yet. The last report on the bold goals for the US biomanufacturing capacity, which is currently undergoing final review and should hopefully be out later. So I think this kind of speaks to the picture on the right side here, where it may appear like the US government is sort of clogging this chromatography column, but instead we're going to envision that as sort of a frit in our chromatography column, and all of these great ideas are flowing through and coming to these final advanced manufacturing strategies. So with that, let's dive right in. So I'd mentioned the National Biotechnology and Biomanufacturing Initiative was really kicked off by an executive order that was signed by President Biden in September of 2022. So what the executive order essentially did was it really launched the NBBI, but it kicked off a whole of government and really whole of bioeconomy discussion and approach to start to really address the key challenges and capitalize on the opportunities that biomanufacturing and biotechnologies afford from an economic competitiveness and well-being landscape for the US population. So on the bottom right-hand side, I've actually taken some text from the original executive order and highlighted 4 critical things that I'd like you all to take away from the talk today. So these are key actions that were hopefully enabled with the NBBI to bolster biomanufacturing R&D, of course, to support societal goals, improve biomanufacturing capacity through adoption of innovative technologies, train and support the necessary workforce to actually make those biomanufacturing technologies run and work, and last but not least, promote standards, common metrics, and develop those systems to really grow the bioeconomy. Now, if you want to take a snapshot of these 4 key actions, these are of course available on whitehouse.gov. But if you compare this, as we'll see later, to the Nimble mission, you'll really see a lot of harmony between what Nimble has intended to do from the get-go and what we still need to do in the greater bioeconomy as a whole. So I think you'll really see that synergy as we progress throughout the talk. So focusing first on the NBBI Bold Goals report. So this was one of the first reports to come out of the NBBI. It went public in March of 2023, and I invite folks to go to whitehouse.gov and read the full report on this. But essentially, this was a global report that involved 5 federal agencies, essentially leading various sections of the report in different topic areas. So climate change, food and agriculture innovation, supply chain, human health, and of course, cross-cutting advances that are sort of those early readiness level technologies needed to foster advancement in biomanufacturing. So each of these individual reports was actually led by an individual agency, but involved interagency coordination and communication in the writing. But more importantly, it involved extensive stakeholder feedback to get the actual academic and industrial perspective to write into these bold goals. What is it that we think as a community we really need to advance biotechnology and biomanufacturing? So when I look at these individual goals, you'll kind of see some common organization in these different reports. And we'll see in the next couple of slides how they're organized. But there's essentially a high-level theme that starts out, a general goal area related to an individual report, and then there's going to be a specific, but not necessarily exclusive example to a specific bioeconomy sector. So when one reads these reports, what I like to tell people is you really want to try to find yourself. So look at the high-level themes and the general goal areas. And I bet that regardless of what your bioeconomy sector may be, in this case, biopharmaceutical biomanufacturing, I bet you'll find high-level themes and general goal areas that align well with the mission in your particular group. You can then look further at some of these contextual descriptions that'll be in the back, and these are bulleted research and development enabling goals that really will help to achieve these bold goals if we can achieve some of these R&D technologies. So in looking at the DOC sponsored report, we can initially start to look at some of these individual themes. So again, you'll see this high-level theme of alternative supply chain pathways via biotechnologies. So when we first wrote this high-level theme, we of course had things like cellular agriculture and industrial commodity chemicals in mind, of thinking about how can we improve and make existing products with biotechnology and biomanufacturing. In the case of biopharmaceuticals, we've been making this with biomanufacturing for several years, but that doesn't mean that you can't look at these individual goals and say, my work is actually accelerating development of biomanufactured products, for example. And if we dig into some of these individual R&D goals, we'll see some common themes. So sustainable and cost-effective capabilities to support large-scale biomanufacturing. Again, seeing that commercial capacity as the end goal of any new technology. Life cycle management of that sustainable biomanufacturing process. We really need to understand the sustainability aspects of these biomanufacturing technologies as we start to integrate them into commercial practice. We'll see that scale up and regulatory science needs are of course mentioned throughout several of these documents, and there's in fact an entire bold goals report dedicated to regulatory science harmonization. But last but not least, in this particular theme area, I wanted to point out that they mentioned platform technologies. Now, this has been something that's been discussed quite a lot in recent months. So talking about platform technologies, for example, for viral vector manufacturing. But I think what's really interesting to note is the Prevent Pandemics Act actually discussed the ability to expedite certain review processes once a platform technology is designated. So I think this is a space to really watch in the coming years, and that we could start to see acceleration of certain of these new platform technologies being adopted and enacted even in a regulatory landscape. Looking at the next theme, biomanufacturing innovation to enhance supply chain resilience. And I think as we look at the first 2 goals, we immediately see some correlations to some of the Nimble programs that I'll talk about later. So the Big Data Initiative really is looking at these predictive capabilities and real-time biomanufacturing process adjustments. So as you can imagine, process analytical technologies, inline, at-line, that can improve attribute relevant measurements will really expedite release of these products in the future. Having real-time feedback loops that can actually change the bioreactor process in real time when potential deviations are noted in the bioreactor process. Starting to enable smart biomanufacturing and biopharmaceuticals, so continuing to integrate the automation, robotics, software, equipment, and data that comes out of the end of those biomanufacturing processes will of course be critical. End-to-end biomanufacturing, these closed systems that enable an input followed by an output of drug substance relatively seamlessly, are of course another innovation that's coming along in many biomanufacturing systems. And last but not least, we see again platform technologies and standards, which we'll focus on much more in the final theme of the Bold Goals report. So of course, last but not least, this was a DOC-sponsored report coming from the National Institute of Standards. One can imagine that standards, both physical, documentary, and material, are critical to the bioeconomy from our perspective. So we can see, looking at some of the R&D enablers, that of course we're talking about data standards here. So ontology, metadata, being able to find, reuse, and really make sure that that data is interoperable with other data systems is going to be critical to really harnessing the power of all the information that we collect about our bioprocesses and our products. My personal interest, of course, is analytical method standards. So we'll talk a little bit about the NIST today and how an advanced measurement infrastructure can actually be kicked off by having a commonly shared physical test metric. And last but not least, I wanted to talk about this last bullet here, translating industry benchmarks, tools, capabilities, and best practices into international standards. So you'll note that Manufacturing USA, of which Nimble is one of the many institutes, is directly called out here. And that's because we have a very large group of industry, academic, and federal stakeholders convened that can really talk together about the standards needs and help to inform the standards development organizations such as NIST as to what standards are really necessary to help continue to move those analytical methods, the bioprocess methods, and new technology into the future of biomanufacturing. So I'd mentioned that when we look back at all of these individual Bold Goals reports that have come out, there's another Bold Goal report that's going to come out in the coming month or two regarding capacity. And one of the things that I think you've seen already from the supply chain report is there are a lot of different elements that have to come together to have a comprehensive system for an advanced biomanufacturing system. So in order to build capacity, we really need an ecosystem, not only to generate an initial ecosystem, but also one that promotes continuous evolution throughout the evolution of both the product and, once the product is approved, continuous change management in the biomanufacturing system for the future. So of course, there's some foundational elements that are absolutely critical. We need a workforce to actually run those bioprocesses and characterize the materials. We need a supply chain, of course, to feed the bioreactor, but also supply chain management to ensure that we don't have drug shortages of critical medicines. Once these bioeconomy foundations are in place, we can start to think about developing new and innovative technologies through manufacturing-related R&D, and having that end goal in mind, translating those technologies through a biomanufacturing readiness all the way to commercial realization of those individual technologies and ultimately those products. So when we consider this entire ecosystem, not only in the context of biopharmaceutical biomanufacturing, but holistically in the bioeconomy, we look back through all of the Bold Goals reports and start to talk to the individuals about biomanufacturing, we see several common themes that emerge. So we see, again, workforce and supply chain. We see pre-commercial and scale-up technologies that are necessary, which could, of course, be test beds, shared spaces to think and create in order to eventually translate those new technologies to true commercial capacity. We see a lot about research and development and translational science, so new and innovative bioprocessing techniques, new analytical methods, and of course, digital architecture to enable a new paradigm in control of these biopharmaceutical biomanufacturing systems. Now, of course, from the standards landscape at NIST, we of course think that there are standard opportunities in each of these individual areas to bring some of them together to help calibrate and harmonize. But ultimately, each of these different bioeconomy sectors have a biological input, they grow some biological material, and we're purifying and formulating some sort of product. So it sort of makes sense from a high level that many of these common themes emerge across all bioeconomy sectors. So this leads us to a continued vision to say that advanced biomanufacturing, regardless of which industry sector it's created in, will likely touch other sectors of the bioeconomy, meaning we can learn from each other not just as a group at Nimble or a biopharma group as a whole, but even from other bioeconomy sectors. And we'll hopefully see some of that later today. We believe then that biomanufacturing will, of course, help to continue to address emerging issues in health, climate, energy, and agriculture. So we'll move on from the bold goals with a very, very brief introduction to NIST. I want to make sure that folks know sort of where NIST comes from and how we engage with Nimble. We are actually a non-regulatory agency within the U.S. Department of Commerce. So our mission really is centered around promoting economic competitiveness in any individual technology and manufacturing area. So we have programs in cybersecurity, food science and nutrition. We even have a fire research program at NIST. And we do all of these individual programs through developing innovative technology and measurement science, but also developing standards or reference materials to be utilized by the industry to both calibrate their test methods and harmonize and innovate new analytical technologies. So the NIST Biomanufacturing Program has been around for a number of years now. And with the introduction of Nimble, we actually now have both an intramural and an extramural program. So the NIST laboratory program has measurement science and standards, as mentioned before. We also have a partnership at the University of Maryland at IBBR. This is where my laboratory is actually located. But we also have our extramural program, and this is, of course, the Manufacturing USA involving Nimble, which we'll talk about in detail today, as well as a couple of other Manufacturing USA institutes in the bioeconomy area. On the intramural side, I want to highlight just a couple of programs and very, very briefly describe sort of how NIST conducts its overall mission. So we have a couple of representative programs that are of interest to this audience. I think, of course, biopharmaceutical biomanufacturing is right in the wheelhouse of Nimble. So thinking about mAbs, cell therapies, gene therapies, and vaccines. There's also a wonderful program in regenerative medicine at NIST as well, thinking about tissues and organoids. And this is more in the sort of Fab Manufacturing USA Institute. But at NIST, at our internal programs, we really perform a lot of measurement science and we develop innovative measurement science technologies. So we have capabilities to measure any biomolecule of interest, from proteins to DNA, all the way to whole cells. These measurements are, of course, for things like primary structure all the way up to higher order structure and potency, as well as online analytical techniques looking at processes and product quality attributes. On the cell side of things, of course, we have several quantitative technologies for cell imaging, flow cytometry consortium to look at biomarkers on the surface of cells, and many other quantitative cell characterization technologies. We combine each of these individual measurement science technologies, and we ultimately apply them to standards that will again help to harmonize measurements in the individual field. So we have microbial pathogen DNA standards. We have next-gen sequencing methods that can really help to understand the accuracy and precision and harmonize analytical methods across sequencing of whole genomes. And the two materials I'll highlight today, the NIST mAb and NIST CHO cell line, which of course are right in the wheelhouse of Nimble. So the NIST mAb is of course near and dear to my heart. This was a program that I started at NIST about 10 years ago. And the intent was to develop a publicly available but representative monoclonal antibody that had all of the biopharmaceutical characteristics that one would expect in a high-quality biopharmaceutical product. So we initiated this program by sending the material out to about 100 collaborators throughout industry, academia, and government. And we characterized this molecule quite extensively. Following that initial book series publication, those methods were qualified at NIST. Values were assigned to this reference material, and it was made publicly available to the community. Since then, NIST and the biopharma community have utilized this material for a number of interlaboratory studies, developing and advancing new and innovative technologies, and really showing what's possible for monoclonal antibody characterization. Fast forward a couple of years, we now have the NIST reference material. Or the NIST research grade test material, excuse me. So this is a CHO material that expresses a non-originator version of the NIST-MAB or the C-NIST-MAB material. And you can imagine that this bioprocess reference material can now be utilized to do the same thing for bioprocess as we did with the NIST-MAB for analytical characterization. You can test new bioprocessing technologies. You can start to understand how new sensors may add additional information to those bioprocesses. And you can start to bear out principles of digital architecture in real time using a real system that expresses antibody at industry relevant titers. So we think that these voluntary pre-competitive materials will again continue to be very impactful in developing new and emerging technologies, and we hope that folks continue to use them for again evaluating, innovating both their analytical methods and their novel biomanufacturing technologies. Moving into Manufacturing USA and Nimble. So I talked a little bit about the Manufacturing USA network in some of the previous slides, but I want to just mention that the Manufacturing USA Institute really does cross every manufacturing area in the United States. So electronics, materials, energy, digital automation. What we're focusing on today, of course, is the Biomanufacturing Institutes. So BioFab USA, located in Manchester, New Hampshire, really focuses on tissues and higher order structural organoids. Nimble, of course, is in the biopharmaceutical space, MABs, cell and gene therapies, and vaccines. And then we have Biomade, which is more of the Industrial Biotechnology Manufacturing Institute. So these are public-private partnerships that are initially catalyzed through federal funding, but sustained through broad industry collaboration. The idea in these Manufacturing USA Institutes really is to focus on industry-wide challenges that no individual company may solve on their own, or may have disparate solutions that, when brought together, offer a much more holistic solution to a biomanufacturing problem. So Nimble, the Biopharmaceutical Biomanufacturing Institute, really focuses, as I've mentioned, on MABs, antibody drug conjugates, cell and gene therapies, and vaccines. And again, if you look at the mission statement on the left here, you can see a lot of harmony with both the NBBI that I discussed earlier, as well as the NIST mission to help accelerate biopharmaceutical manufacturing innovation through adoption of innovative technologies. I think that adoption of technologies is really critical. Support the development of standards to help to inform the standards developers such as NIST what standards needs are really necessary to advance the biopharmaceutical community. And of course, Nimble has a very strong program in training a world-leading workforce to help to manufacture these individual products. So Nimble's been around since about 2017 when they were initiated, and has since been renewed. And then under the leadership of Kellen Lee, we've actually grown this institute to over 200 members now, which really is a fantastic cross-section of the biopharmaceutical community. And we'll see in the next few slides the innovation that actually has been achieved in the past few years. So as you may know, Nimble actually delivers its mission through a couple of different pipelines. So on the right-hand side here, we have the member-led projects. So this is a typical sort of RFA process where there's a project call with specific kind of high-level needs of the biopharmaceutical industry. And typically small teams of 2 or 3 organizations, often an academic and an industry partner working together, will provide a solution to a manufacturing problem or an analytical characterization problem. So these are usually rather discrete projects, about a year in time. They're co-invested between the partners, and they really, again, try to specific on a very targeted problem or a targeted assay, targeted solution for biomanufacturing or analytical characterization. It was realized later that there were some challenges, again, that were sort of big picture, that this project call mechanism really didn't quite fill the bill. So this is where the institute-led projects were initiated. So these are much larger projects that are, again, initiated with very broad industry input and industry leadership. They're multi-year programs, but really intended to tackle those big challenges that no individual company is going to solve by themselves, or again, a problem that when brought together, each of the different pieces from multiple companies can really provide a wonderful solution to grand challenges or some of these bold goals in the biomanufacturing area. So starting off with the member-led projects, I just wanted to point out to folks that there is a website online that you can go and search each of the projects that have actually been completed, as well as those that are ongoing, and get a paragraph summary or even more about some of the outputs of these individual projects to really understand what some of the impact and what some of the technologies that have been developed at sort of a readiness level of 4 or greater. So here I just typed in a keyword, gene therapy, and of course this brings up my personal favorite. We all have to have one. This was actually a project that my lab, Pfizer, NC State, and BTech down at NC State worked on together to develop some new process and analytical characterization methods for AAV. And I want to highlight this because it really shows the impact that Nimble can have bringing together industry, academia, and government agencies to, again, work on a problem that all of these organizations are interested in. And I'm happy to say we've got a couple publications coming out. One was accepted just last week and another in a couple of weeks from this project, again, really demonstrating that ability of Nimble to bring people together. Another project example that I wanted to discuss is pandemic response. So as you all know, we had a pandemic a few years ago. We're now coming out of that pandemic due to some wonderful therapeutic modalities. But in the early days, it was recognized that we really needed to invest in new research, development, and test beds to help prevent the current coronavirus or prevent future pandemics, prepare for them, and respond to the current coronavirus pandemic. So Nimble quickly mobilized its community and came up with a wonderful group of project proposals that were proposed to NIST. Nimble very rapidly executed these individual programs, and we now have a dossier of 32 projects that are kicked off. Roughly half of those are now complete. The rest are aimed to be completed about within the next year. And these focus, again, as you would think, on many areas that are critical to a pandemic response. So in the upper left-hand corner, we're building test beds that are critical infrastructure that can be shared spaces in the community to develop innovative manufacturing and quality testing of these medical countermeasures. In the right-hand side, we've had several projects that are looking at advanced medical product manufacturing. So this included things like developing prototype LNP mRNAs to optimize things like lyophilization and other drying technologies to try to minimize that cold chain necessity for that modality type. There were projects that were looking at alternative cell systems to rapidly express monoclonal antibodies. And there was even some on continuous purification of mRNA as it's produced. So quite a broad dossier of projects in that individual scope side. Of course, a pandemic response requires testing and the ability to test for emerging variants. So there were also several projects that were funded on rapid development of coronavirus antigens for diagnostic assays, some new and innovative diagnostic testing assays, including miniaturized PCR, and several other projects that were targeted at testing for these emerging variants. And last but not least, I'd mention one of the big facets of Nimble is workforce development. So we also were able to expand the workforce development portfolio, including many pandemic-related biomanufacturing courses. So another thing that I like to really point out about the ART portfolio is, as any good pandemic response would have, we have a very diverse type of modalities that we're looking at. And you can see that there are projects funded across lipid nanoparticles, RBD spike proteins, MABs, and other types of modality-mediated vaccines. So a really nice diverse portfolio of projects that was funded. So now moving into some of the Nimble-led programs. So we'll start off with NMAB, which was a really fun project that I actually learned, more than I was ever hoping to when I went through the process and working with the large group of people that came together from the NMAB, led by Gene Schaefer. So this was really an update to the AMAB case study, which, if you all remember, in 2004 came out, and this was really sort of an archetypal test case to guide quality by design of monoclonal antibodies. Well, the NMAB updated this case study to continuous integrated drug substance processes, and actually had two individual processes that were considered, both with and without a batch pool tank in the middle. So what the NMAB case study really did was, again, generate an archetypal case study to look at process design, characterization, demonstration, and validation control. So it goes through all the steps and considerations that one would typically go through in developing an end-to-end continuous integrated drug substance process. So this both helps to reinforce the vocabulary that can be used in this case, but also with the process descriptions and sort of how one would approach this continuous end-to-end, it starts to develop a strategy that we can potentially then test out in real time with actual test beds and actual demonstrations. So I want to call out as well that there's a wonderful chapter at process deviation, again, one of my personal favorites, talking about lot disposition when there is a deviation that occurs, essentially how one will batch different portions of a continuous integrated biomanufacturing system. So I just want to point out this is a wonderful read for folks that are looking to get into process control of their integrated systems. So biomanufacturing readiness level is something that I've sort of alluded to a little bit. And some of you that may have had some past experience with other manufacturing areas have often heard about the DoD's manufacturing and technology readiness levels. So this essentially tells or is a maturity metric to understand essentially the technical readiness, quality readiness, and operational readiness of a new technology towards implementation to that end goal of commercial manufacturing. So what we've done with the biomanufacturing readiness levels is started to take some of those concepts from DoD and applied biomanufacturing language to help assess the technology maturity with the end goal of a commercial biomanufactured pharmaceutical product at the end of concept realization. So this BRL, of course, is a common vocabulary, so it's very useful in talking amongst colleagues to say, this is where my technology is at, to help define its maturity level. It's also a wonderful tool to do a self-assessment to say, maybe my technology readiness is at a 5 or a 6, but I'm kind of lacking behind perhaps in quality or operational readiness. Maybe I need to work on facility or workforce fit to ensure that this technology can truly be adopted in a commercial setting. So overall, we really hope that this sort of framework will help folks to both translate their technologies to that ultimate implementation in a commercial setting. The next test bed is the process intensification program and test bed. So this is a fantastic demonstration of, again, the biopharmaceutical community coming together around a grand challenge to say, can we set up and demonstrate a 50-liter scale continuous integrated bioprocess test bed? So I'm, of course, ecstatic to see that this test bed is actually using a Nimble CHO cell line, producing the same NIST mAb non-originator molecule at, again, industry-relevant titers as the test case to actually bear out the principles of this continuous integrated test bed. So what you can imagine is now that this test bed is available, and we have folks from numerous companies coming together at Nimble headquarters to work on this test bed, we can now start to bear out some of those principles from the NMAB case study, lot disposition, etc. But we can also think about this as a playground to sort of take new and emerging technologies, whether this be sensors or new unit operations, change them in and out of this test bed, and really understand from a comparability standpoint how those might affect manufacturing of this common drug product. Again, doing all of this in the pre-competitive space. There's several other arms to this program that I can't get into in too much detail, but I want to mention that there's a very strong program in flexibility, looking at configuring sensors in and out, being able to do hot swaps in the middle of a manufacturing run, and a wonderful program in sustainability, doing life cycle assessment and putting together tools to really understand the sustainability of single-use biomanufacturing. The Big Data program is another program that has had wonderful success in bringing together the community, and I think is really trying to tackle one of the biggest challenges in biopharmaceutical manufacturing, which is now that we have the capability to actually collect all of this data, how can we effectively use that to better our process, ensure we have high quality product, and essentially move product to patients faster? So the Big Data program has an ultimate goal of generating a lights out adaptive control of a critical quality attribute, targeting initially glycosylation, but using other new and innovative PAT as well. Of course, looking at inline, online, and antline sampling techniques, again, to get that quality attribute information to feed back to that adaptive control. Of course, this requires mechanistic models of the biomanufacturing process to actually help make those adaptive control decisions, which of course requires a rich data set to feed those individual models. And at the base of that, you of course need a way to establish the data storage, be able to exchange that data, and be able to essentially reuse that data to again effectively build and actually actionize those individual mechanistic models for adaptive control. So you can see that this sort of foundation of data is building forward into each of these individual components of getting towards that lights out adaptive control. I'd say we've really had some fantastic movement in getting some of these PAT analytics set up down at BTech in North Carolina. And we have a wonderful group at NIST that's actually contributing as well to developing some of the ontologies and metadata platforms to help build out this overall big data program. So looking for wonderful things coming out of this program in the coming years. So now for a little bit of forecasting as we close out the presentation today. I hope everyone comes back and joins us next week because there's going to be a much more in-depth presentation on the new viral vector manufacturing and analytics program. So this was actually launched just a few weeks back, and you'll see from the mission here that one of the big milestones is to develop a robust, open and shared platform for manufacturing of AAV-based gene therapy vectors. So the mission really is to promote access to this very critical gene therapy, lower the cost of developing gene therapies for rare diseases to, again, ensure that we can treat more of these indications that are a critical unmet need in the United States at the moment. So as one would expect with any Nimble program, there will be a strong workforce training element to this program as well. And again, I invite all of you back next week to the Nimble webinar series to hear more about the viral vector manufacturing and analytics program. The vaccine manufacturing and analytics program also just recently kicked off. The program leads held a wonderful session at the Nimble headquarters just a few weeks ago to start to vision what this program is going to look like, how it can draw off some of the experiences from the American Rescue Plan, and begin to really establish a center of excellence around manufacturing and analytical characterization at Nimble, again, drawing on broad expertise from the community and broad participation from all of the Nimble group. As we continue to forecast, the last slide talking about Nimble programs is workforce development. So I hope that you'll come back in 2 weeks and join us to hear much more about the workforce development program. So I've mentioned at a high level that workforce is really important. There's several training programs going on at community colleges and colleges that have been funded by Nimble. There are groups that are starting to harmonize their programs to have common credentials amongst individual programs for training of these individual folks. And there are several programs ongoing at Nimble to make sure that folks are aware of the types of biomanufacturing careers that they could potentially have in this particular industry at a very early stage in their training. I also want to highlight that there's directories of all of the training that's available. So of course you can go to the Nimble website and search training there to get an understanding of what some of the training projects that have been funded. But also there's now a digital directory with the DCVMN that Nimble co-developed with PATH, the Bill and Melinda Gates Foundation, and DCVMN that has a comprehensive listing of all of the training programs that were identifiable. So you can go and find training programs, whether they be Nimble funded or otherwise, to ensure that you're getting the right training for your folks in your biomanufacturing needs. So as a summary and conclusions today, of course, we're going to come back to that opportunity space in that pot. What I hope I've shown you today, at least, is in the area of mAbs, RNA vaccines, cell and gene therapies. The U.S. strategy that we've laid for the bioeconomy really includes the biopharmaceutical biomanufacturing industry as a critical component. And the fact that this really is the longest standing bioeconomy sector, I think is really critical to know that other bioeconomy sectors can learn from the innovations and at times potentially the hardships of this industry to make sure that we begin to grow a very robust bioeconomy that includes many of those other sectors in the future. Hopefully this U.S. strategy helps to begin to lay that foundation, looking at the bold goals reports and looking forward to out years where we have more funding and more roadmaps to really guide where the U.S. is going to invest in these different industry sectors. I hope I've also shown today that measurement science and standards really support the biotechnology and biomanufacturing industries through both supporting harmonization of test methods, calibration of test methods, and developing new and innovative technologies. Now, this section of the talk was very sparse. I could have spoken for a couple of hours about all the wonderful programs that are going on at NIST. So I invite you to definitely reach out to me if you're interested, and I can connect you with the appropriate division at NIST. Feel free to visit nist.gov. There's some really wonderful descriptions and explanations of those programs, and hopefully you check those out and see the great connection between NIST and NIMBL in all of these areas. And last but not least, where I spent a good part of my time, I really tried to give a very high-level overview of all of the programs that are going on at NIST. I wasn't able, of course, to get into the individual details of those, but what I think you can really see from this is that Nimble connects people, ideas, and technologies to carry out their missions. So Nimble really is a team sport that relies on the input from the entire community. And I think that's what really makes the innovations that come out of Nimble so great, is they have such a broad input and broad expertise to really try to develop the best solutions to an individual biomanufacturing challenge. So with that, there are many people to thank. There are many people that I'm sure that have been left off of this list, but as you can imagine, all of the work presented here was not done in my office here in Gaithersburg, but rather there's a large group of people from OSTP that contributed to several of the Bold Goals reports. I specifically want to call out the folks from DOC, Chris Zakel, Shenglin Gibson, and Katie Buckle for their work on the DOC-initiated supply chain report. Really was a pleasure working with them on that report. The folks from Nimble, of course, leadership from Kelvin Lee, Gene Shaper, and the rest of the Nimble Fellow team that are really guiding and leading these industry-led programs with various industry members. Of course, there are several support staff, both on the technical administrative side, that help these webinars run, and behind the scenes at Nimble really make it a great place to work. And last but not least, the folks at NIST. Kelly Rogers is the program manager for Nimble and really has been on the federal side moving this program for so many years and is my division chief now in OAM, really helped to guide and integrate into the technical program manager role. Mike Tarloff is my division chief at MML and really helped kickstart the NIST MAB and leads the biomanufacturing program at NIST, and many other folks at NIST that have helped both on the Manufacturing USA and NIST side, as I've mentioned before, really as a team effort to produce these innovations. So with that, we'll close out, and happy to take any questions.
Yeah, thanks, John, for that very nice overview. For those of you on the call, if you have a question, you can enter it via the chat. And enter your questions via chat, and then I'll relay them to John. So there's a few questions here. One is for the AAV manufacturing program that you mentioned. Is this program agnostic of how AAVs are manufactured? For example, mammalian versus insect cells versus plants.
Absolutely, yeah. So you'll definitely hear a lot more details on that next week, but I can share that we had a couple of workshops last year, actually, one process and one analytical. And this was something that was discussed quite a bit in those individual workshops. So initially, we're going to target on a mammalian culture system, so HEK, to develop that open source project. But that's not to say that it wouldn't be of interest to understand and do a project to look at the differences between these various characterization systems. But HEK will be the initial focus.
And a related question, first of all, thanking you for a very informative presentation. Are there any plans in progress in developing and establishing lentiviral vector standards?
Absolutely. So I would feel free to reach out to me. So Shenglin Gibson's group at NIST is actually working on that. They published a paper a few years ago, actually, with a gene copy editor, a gene copy number standard for lentiviral vectors. So the group at NIST is working hard on Lenti, Adeno, and other viral vector standards. They actually had a workshop a couple of weeks ago as well, kicking off a gene editing consortium as part of their flow editing or their flow cytometry consortium. So certainly for folks interested in the lentiviral standard, reach out to me and I'll make sure that I can make the appropriate connections on the NIST side.
Yeah, we had one general question, which is in a way more nimble, but the challenge of providing the cost share. And I'll give my thought about that. But first of all, I mean, because the goal is, NIST being part of Department of Commerce and Nimble, the goal is to be industry led. So we want to be sure we have projects that are of interest to Nimble and hence the relatively high demands for cost share. But I'll ask John to give his perspective on that.
Absolutely. I think that you're right on, Barry, that the cost share really helps to ensure that the project is something that's of high value to the industry and also is at that biomanufacturing readiness level that is getting closer to that commercial adoption. So I think that's certainly one rationale for the cost share.
And then we have a question about workforce development as the foundation for a resilient supply chain. Can you speak to NIST efforts or intention towards any skills standards for this sector? This question comes from an organization focused on skill-based hiring adaptations for companies. So,
Absolutely. It's an interesting concept to think about the type of training for supply chain systems. And, I think there are definitely some programs in other areas that are looking at, for example, techno-economic models would be another Department of Commerce institute, for example, or another Department of Commerce agency thinking about techno-economic analysis and understanding supply chain pathways. There was a couple programs that were proposed that we didn't quite go through with at NIST and NIMBL under the American Rescue Plan, but I think training in this area is definitely interesting of whether that's a digital modeling type training. Are we thinking about, folks from that aspect or supply chain logistics and shipping? There's a lot of different, I think, areas that one could think about for training. So great question. I'd love to chat more with you on that and come back perhaps in a couple of weeks to hear John Balkunis. I'm sure he would have some additional wonderful answers. He's our workforce director at Nimble.
thanks. Yeah. And then there's a general question which I'll hopefully help answer in a few minutes, but it's for projects that are completed, like the AAV analytics project, where can one find the results or output? So I'll come to that in a minute, but I'd first like to thank John for the presentation, and maybe you could go to the next slide, John.
See, we're having some technical difficulties now, of course, but there we go.
Yeah, so this relates to that question about following following a particular project, and you can get email alerts on programs if you're a Nimble member. You can get email alerts on programs and projects you would like to follow, and so this gives you a little description of how that occurs. And you can, if you have a followed project, you will get an email alert as to when there's new updates on that project. And to view the updates, you go to the Updates tab on the project page. So there is a way of doing this. There's a bit of a time delay between finishing the program and being able to access the results. So you just have to be patient with that. So John, can you go to the next slide? So I do want to again thank you, John. Been a great colleague in terms of this journey we're on at Nimble, and the and this is in closing. But the survey reminder: attendees will receive a survey link by email shortly after the webinar. But you have to, and then if you complete the survey, you would get access to the webinar recording. And John mentioned we have a Nimble-led program in viral vector manufacturing and analytics. The webinar is next week, November the 15th, with Tim Charlebois and Eric Hacker. So looking forward to that. And then the very active workforce development program at Nimble, led by John Balchunas, will be November 29th. So these are two upcoming webinars. I hope you can tune in for those. And then the next slide, John. And just to get this on your radar, we had a really good national meeting in 2023. We learned a lot from that, but basically, I think the basic concept behind the meeting was really good. So we'll probably be very similar for 2024, at least in many respects. We'll be focusing on Nimble programs, and it was a really great event of over 300 people in person and a number of people remote, but very, very vibrant and energetic meeting. So I'd like to thank all of you for tuning in, and especially John for giving a presentation, and look forward to all of you joining, helping Nimble on its journey. This is our organization. We rely on our members to drive the programming and to drive the investments. So it's been an exciting journey so far, and look forward to continuing. So thank you very much. Thank you. Bye-bye.